I don't think so.
No, you're good.
Okay.
All right, I'm going to switch the room, and as soon as you see yourself up there, you can start.
Hello, we're live at Cold Spring Harbor. Welcome to our fifth Gramene webinar, and thank you all for joining us. Last month, my webinar, I described an overview of the Gramene genome browsers. This week, I'm going to focus in a little bit more on gene trees and exploring phylogenetic gene trees. I want to first point out that all of the resources that I'm describing are available both on Gramene and at the website of our collaborator at EBI through this URL, plants.ensemble.org, and all the video that I'm going to show you actually came from Gramene.org, but they represent the same thing. Okay, so I'm just going to give an overview first on the browser and then go into more depth in the Gramene gene trees. So Gramene adds value to plant genomes. Basically, we take accessioned published genomes plus community annotation from various primary sources, and we do standard annotation pipelines to look at various functional annotations, and we do comparative analyses, including whole genome alignments and gene trees and synteny mapping. And then we present these as a variety of different visual displays. Plus, there's programmatic access that you can use. So before going on, I want to thank actually all of the community genome projects. All of the data that we show actually came from publicly funded sources. And currently, our species list is now up to 38. And so this is the Gramene genome portfolio. So we have 38 species, and this is the species tree. So this comprises 20 monocots with heavy representation of oryza and related rice species, plus other cereals in the grass family. But we also have banana. We have 12 core eudicots and 1 basal angiosperm, Ambarella. And we also have 5 lower plants. So I'm going to focus in on comparative phylogenomics today, which I gave a brief overview of last time. 2 areas that we do are whole genome alignments, and I'll touch on that a little bit today, but mostly I'm going to focus on the phylogenetic gene trees. So this is just some background on the protocol. Essentially, all of the protein sequences from all of the species annotations are clustered to generate homologous families. And then there are 5 different tree building methods, which are listed here. And then a program called TreeBest is used to build a consensus of these 5 trees. And once that consensus is built, various analyses are performed in order to infer orthologs and paralog genes. And we output visual display of gene trees with that are annotated, the nodes are annotated blue are speciation nodes, and so those are the products of speciations, and that's what defines orthologs. And red are duplication nodes, and that's what defines paralogs. In addition, the nodes are classified by their taxonomic origin. So for example, this node here is at the common ancestor of mouse and human in this example. And there are also various complicated relationships. Orthologs can be one-to-one, where there's just a single ortholog in each species, or there can be multiple orthologs in one species and only one in another, or there can be multiple to multiple. Okay, so last time I described a use case on domestication genes, and I'm going to actually go back to this use case because it's a very interesting gene family. So The SH1 locus is the shattering 1 locus, controls seed dispersal in grasses. And this actually is a domestication gene. Early farmers selected for plants that did not disperse seeds, so that when they went to harvest the grain, they were able to hold on to it without it dispersing. And this turned out to be a mutation in the gene called SH1, which is a YABBY transcription factor. In Asian rice, there's a 4 kb insertion that limits the expression of that gene compared to the wild-type gene. And in addition, it was found in sorghum that this gene is disrupted. So this is a case of convergent evolution or parallel evolution. And so the question here in this use case is, if we look at other domesticated cereal grains, what do we find? In this case, we're looking at African rice, which is a separate species of rice called Oryza glaberrima. So that was recently sequenced by Rod Wing's group, and in addition, they have sequenced Oryza barthi, which is the progenitor of African rice. And if you look at the tree here, you can see the rice gene, which is labeled as YAB2. It's known, it has various different names. But in Asian rice, in our trees, it's called YAB2. And you can look at the structure of this tree, and you can see example genes from many other Oryza species. We have at least half a dozen species of rice in here. Whoops. And we see the gene as well in Oryza barthi, but it is noticeably absent in glaberrima. There's no gene in glaberrima. So we can then actually go to the whole genome alignments, and we can compare the alignments of, this is Asian rice, and above here is Oryza glaberrima, and down here is barthi, and these brown marks show genes, and these blue lines link together orthologous genes, and these green ribbons show where the whole genome alignment is. We can see that there's a very large deletion in Oryza glaberrima. And this was found, and therefore the SH1 gene is in fact missing in Oryza glaberrima. And this finding was actually recently published by Rod Wing's group. So I'm going to expand more on the YABBY gene family, just because it turns out to be a very interesting family. In Arabidopsis, there are 6 genes, and several of them are known for their developmental phenotypes when mutated. In rice, there are 8 genes, and in addition to the shattering 1 locus, there's also a phenotype called drooping leaf of a different member of the YABBY gene family. And I'm going to now switch to a movie demo, and I'm going to demonstrate navigating gene trees, how to browse and get to orthologs and paralogs of genes, and also looking at the alignments. So this is a 10-minute video. Okay, so right here, we are on the location view of the browser, and this is the rice browser. And we can scroll down and click on the YAB2 gene, which is the shattering 1 locus. And if we click on the gene itself, that'll take us to a different page called the gene page. So you can see along the top there are several tabs. One is the location view, the other is the gene view. And each page has a set of menus that are relevant to that page. Here on the gene page, we have the gene trees and the alignments, as well as the ortholog and paralogs. So I'm going to click now on gene tree, and this is going to bring up all of the gene family for YABBY genes in plants. And at the top here is an overview of this family. There are 289 genes across all of the 38 plant species that we have. And it also lists the number of orthologs and paralogs, duplication nodes and speciation nodes. So now all of the genes that you see here can be clicked on, and when you click on them, you get additional information, and it also enables you to go back to the location view. Here, you can also click on internal nodes, and for example, if you click on an internal node here, it tells you what taxon it's from, and also you can do various other options. Here, I'm clicking on the Newick tree format, and this will actually give you a text version of the tree at that node. Okay, let's see. Okay, we're back to the tree here. Now, most of the nodes here are collapsed. You can see that the tree is actually very compact, and particular taxa are collapsed here. So, but we can expand individual taxa. So here we've just clicked on a node, and we are expanding that node, and now you can see whereas that node was once represented as a collapsed sort of triangle, now you can see all of the genes and the species in that node. There are several other options. One is you can view all of the paralogs to the gene that you're interested in. So that's what I've just done here. At the very bottom, clicked on View All Paralogs. And in blue, now we see all of the rice paralogs of YAB2. So the red shows YAB2 itself, the blue shows the rice paralogs, and you can see that there are 8 genes total in rice. Another option is to view all of the duplication nodes, and so I'm clicking now on duplication nodes. And this will show all of the nodes that are red, that represent duplication nodes that result in paralogs, are expanded in this case. So you can see where all the duplication events occurred. So I just saw one in maize. There's a lot in bananas, you'll see later. There's a lot in soybean as well. So I'm just browsing down, checking this out. That's a bunch in maize, in banana. Okay, and then finally you can click at the right there to get a fully expanded tree. So this will show all 289 genes in the tree. So as you scroll down, it'll show that. And then I want to point out on the right-hand side is actually a schematic diagram of the multiple sequence alignment. So the green areas on the right-hand side represent where the sequences, where the protein sequences actually align. Okay, there are other ways you can configure this image. So if you click there, this menu will pop up, and you can actually have more fine-grained control over what nodes are collapsed and what aren't. You can actually look at the other types of trees that were built. So I mentioned before there were actually 5 different trees, and we're showing now a consensus. You can actually look at an individual tree. For example, if you wanted to look at the neighbor joining tree that was done at the amino acid level. Okay, here is another really nice feature. So all of these genes, as I mentioned before, are annotated functionally, and we have 2 different ways. One is Go, and the other is Interpro. And you can bring up this menu, and it'll show you what the annotations are for the genes in this tree, and you can click on one of them, and it'll show you which genes are actually annotated that way by highlighting in green. And so what I just showed you was clicking on the Interpro domain that represents the YABBY protein domain. Okay, so now let's say you want to retrieve orthologs. On the right-hand side of the menu, you can click on orthologs. In parentheses, it says that there are 38. So that means that there is a list of 38 genes across all of the different species that are orthologs of the rice YAB2 gene. And you can get various types of information. They're all listed here. You could download them as an Excel spreadsheet. For each one of these, you can actually look at their alignment at both the protein level and cDNA level. So here I've clicked on an alignment, and this is going to show the alignment between the rice YAB2 gene and the brachipodium. And also, as I showed last time, you can also use this to launch back into a location view where you're comparing the two regions of the genomes. And so here, we're now going to compare the region in rice that contains the YAB2 gene to its orthologous region in brachipodium, and that's shown here. And I'm going to zoom out in order to get a larger view. And this will load a much bigger view. We're basically zooming out, and it'll show all of the genes in the region and their links showing the synteny, levels of synteny. Now you can see that brachipodium is inverted relative to rice, and you can flip the strand if you want to get a more comprehensible view. Okay, now I'm going back to the gene tree. And I'm going to demonstrate also, just as you can get orthologs, you can also click on paralogs to get within species paralogs. So there are 7 paralogs in rice that are related to the YAB2 gene, and that's shown here. There are various, in addition to looking at individual alignments, you can also do one click and view all of the alignments at once. So that's shown here, each of them shown in order. Now let's see, what am I doing next? You can download the multiple sequence alignments in a number of useful formats. So Clustal W, FASTA format, Mega format, MSF, Nexus, et cetera, et cetera. So you can get this menu here. You can choose which format you like. Click on that, and that will download for you. And now you can also, in addition to looking across 2 species, you can actually look within a species. So here I'm going to compare the YAB2 gene to one of its paralogs. So this shows a browser view of rice chromosome 3 that has the YAB2 gene with rice chromosome 12, which has one of the paralogs, one of the duplicate copies of the YABBY gene. And you can actually see, in addition to the YABBY2 gene, there's some residual duplication of other genes in that region as well, probably due to the whole genome duplication in grasses. Okay, that's the end of the movie demo. So going back to the slides, I'd like to just point out some of the, if you actually spent more time examining that tree and adjusting the various views, here are some interesting observations. If you click on one of the Ambarella genes and then select View Paralogs of Ambarella, Ambarella is a basal angiosperm, so it lies at the base prior to the split between dicots and monocots. And so this gives a pretty good indication of what the ancestor looked like. And you can see that there are actually 5 genes, YABBY genes, in Ambarella. And what's interesting is that 4 out of these 5 are actually associated in clades that exist with extant monocots and dicots. So 3 of these are found in monocots and dicots. 1 of these is found just in eudicots, according to this according to our tree, and one is unique to Ambarella. So I want to point out here that these are our observations that we're making with the data. These are hypotheses that we're building. If we wanted to really form firmer conclusions, you would want to do some additional analysis. But at least this is giving us some interesting tidbits of what might be going on in this gene family. So maybe there were 5 ancestral YABBY genes. In grasses, there were additional duplications that are common to all of the grasses that led to 8 final copies. And then if you actually look further, you would be able to find individual species-specific duplications as well. One thing that I found really fascinating when looking at this is if you look at banana, so here just the banana genes are being displayed. There are actually 25 genes in banana, so, and those are shown in blue. So banana is a monocot, but it's not a grass. So what we see in banana represents all of the evolution that occurred in monocots after it split from the grasses. So we can hypothesize here that there were a very large lineage-specific expansion in monocots after the split with grasses. Or they could be specific to banana. We'd have to look at more monocot species to determine that. Also, among different species, we found a lot of species-specific expansion in soybean. So this is very interesting given that YABBY is a transcription factor. There are several known examples in the literature of how they're involved in development, both in flower development as well as in leaf polarity development. So the fact that we see a lot of expansions and differences in species could lead to some interesting future research. Okay, I'm going to end here, and I just want to again acknowledge our collaborators and also give thanks to the rest of the Gramene team and our collaborators. And I also wanted to thank all of you for joining us for today's webinar. So, are there any questions? Can someone read that for me? Oh.
Just says thanks.
Oh, okay. Okay, thank you. Well, I was